Hello, I'm Dr. Michael Lee, and I will be guiding you through this self-learning module in enzyme kinetics. This session serves as the basis for much of what we will cover and discuss in the class on pharmacodynamics, so it's important that you develop a clear understanding of the key points that I'll emphasize here. Briefly, we'll discuss general properties of enzymes, kinetic properties of enzymes, enzyme inhibitors, allosteric enzyme regulation, and finally isozymes. Our learning objectives for this session are define activation energy and explain how it changes during enzyme catalysis, describe the role of cofactors in enzyme-catalyzed reactions, define the terms Km and Vmax which are used in the Michaelis-Menten equation, distinguish allosteric kinetics from classical Michaelis-Menten kinetics, distinguish competitive and non-competitive inhibitors by their effects on Km and Vmax, define isozymes and discuss the basis of their diagnostic value, and finally, by the conclusion of this session, we should be able to describe the means by which enzymes are regulated. So first, let's start off with the general properties of enzymes. As you have probably already learned, enzymes are highly efficient protein catalysts, which are not consumed during the catalytic cycle. Enzymes generally have characteristic locations inside tissues and cells, and they contain a specialized region known as an active site. The active site contains amino acids that are involved in binding to the substrate, and also contains catalytic amino acids that are involved in executing the catalytic reaction. There are two different models that have been proposed to explain how enzymes interact with substrate. There's the lock and key model, where the enzyme's active site resembles the topology of the substrate, and they interact together like a lock and a key. And then there's the induced fit model, where the substrate induces a conformational change in the enzyme so that both molecules fit together. There are several characteristic reactions that enzymes catalyze. These include oxidation-reduction reactions, for example lactate dehydrogenase, which converts lactate to pyruvate in glycolysis by adding a hydrogen ion. Remember, reduction is a gain of an electron and a proton, whereas oxidation is loss of an electron and a proton. Another type of reaction catalyzed by an enzyme is group transfer, such as phosphorylation. For example, phosphorylation by a kinase like ABL. Enzymes also catalyze hydrolysis reactions, such as the enzyme maltase, which hydrolyzes maltose to 2 units of glucose. Enzymes such as lyases can cleave carbon-nitrogen, carbon-oxygen, or carbon-sulfur bonds without oxidizing, reducing, or adding water. The enzyme aldolase, which is involved in glycolysis, is an example of a lyase. Another characteristic type of enzyme is an isomerase. For example, glucose 6-isomerase converts glucose 6-phosphate to fructose 6-phosphate in glycolysis. And finally, we have ligases. Ligases such as DNA ligase form covalent bonds between nucleotides. Many enzymes rely on cofactors. These can include inorganic cofactors such as metal ions, which stabilize the enzyme, assist in substrate binding, and also assist in catalysis. Or cofactors can include organic cofactors, also known as coenzymes. These are generally heat-stable organic molecules that are derived from vitamins, and they assist as carriers of electrons or chemical groups in group transfer reactions. Enzymes that use cofactors are referred to as apoenzymes in the absence of the cofactor, and they're inactive, whereas when they're bound to the cofactor, they're referred to as holoenzymes, and this would be the active form of the enzyme. So how do enzymes work, and how do they catalyze a reaction? Well, it turns out that enzymes actually lower the activation energy of a reaction. For our purposes, activation energy, or EA, is equal to transition state energy, also known as delta G prime. The rate of a reaction is inversely proportional to delta G prime, or E sub A. So the larger a delta G prime, the slower the reaction. The smaller the delta G prime, the faster the rate of a reaction. But you're probably asking yourself, what is the transition state? Well, it's actually the first step in a reaction, where a substrate goes from being a substrate into a transition state where it's bound to the enzyme. So in essence, the enzyme is actually altering the structure and the conformation of the substrate, causing it to look like a hybrid between substrate and product. It's this transition state intermediate that has the highest amount of energy in the reaction. Therefore, enzymes lower the activation energy by stabilizing the transition state. Let's look at this graphically. If we plot out the rate of the reaction, or reaction progress, versus free energy. And we look at the amount of substrate compared to the amount of product. We see that an uncatalyzed reaction has a characteristic activation energy. This hump is the T state, or the transition state. And this difference represents the activation energy, or delta G prime. And again, this would be the uncatalyzed reaction. A reaction that's catalyzed by an enzyme, which lowers the activation energy, would look something like this. You can see that the difference in activation energy for this catalyzed reaction is smaller. So by lowering the activation energy, enzymes actually accelerate this reaction and push substrate to become product. So another way to look at a reaction rate is to look at it from a physics perspective. In other words, if we look at the change in the amount of product over time, this actually yields a velocity. So in this case, if we look at time versus product formation, what we see with an uncatalyzed reaction is that the reaction rate is very slow. However, an enzyme catalyzed reaction would have a characteristic hyperbolic shape. Importantly, a key determinant in the shape of this curve is substrate concentration. And we can see this if we plot out product versus time at varying substrate concentrations. At low substrate concentrations, progressing to higher substrate concentrations, we see changes in the shape of the curve. And from this we can extrapolate a velocity. So the rate of a reaction and the velocity are influenced not only by enzyme, but also substrate concentration. So in addition to substrate concentration, the rate of a reaction is dependent on rate constants and also intrinsic properties of the enzyme, which we'll look at in a moment, but are known as Vmax and Km. So if we look at a reaction, we see that the enzyme plus the substrate undergoes conversion into a transition state, which we talked about a moment ago. And so we have an enzyme-substrate complex. This is a reversible reaction that's governed by two different rate constants, K1 and K-1. And from here, the enzyme-substrate complex can then proceed to enzyme and product. And this is governed by a rate constant called K2. There's two different assumptions in this reaction. One is that the slowest step is the conversion of ES to EP. So this is the slowest step. The other assumption is called the steady state assumption, and this states that the rate of ES formation is equal to the rate of its breakdown. So taking these two assumptions into account, in its most simple form, we can say that the velocity of a reaction is equal to K2 times ES. But as I mentioned a moment ago, there are some other variables that we're not taking into consideration here, and these are intrinsic properties of the enzyme, Vmax and Km. When we take these into consideration, we get the Michaelis-Menten equation. V is equal to Vmax times substrate concentration over Km plus substrate concentration. Let's look at this graphically. But first, what are Vmax and Km, and what is their significance? Well, let's draw out that equation again. And you can see here that Vmax is the rate obtained when all the active sites on the enzyme are filled with substrate. And this is really an index of catalytic efficiency. This is something we see at the highest concentrations of substrate. Km, on the other hand, is the concentration of substrate required for half maximal velocity. And this is really an index of the affinity of the enzyme for the substrate, so that we can say with some confidence that a large Km means that the enzyme has a low affinity for the substrate, whereas a small Km means that the enzyme has a high affinity for that substrate. So now let's look at this graphically. You'll see that this looks very familiar. We have substrate concentration on the bottom, and reaction velocity on the side. As substrate increases, the rate of the reaction increases to a point which we call Vmax. Km, on the other hand, is the half maximal velocity. So at high substrate concentration, when S is greater than Km, the enzyme is saturated and it's at maximal velocity. It literally is churning out as much product as it possibly can, and this is termed zero order. And therefore the reaction proceeds in a manner that is independent of substrate concentration. At this point, we're now beholden to the activity of the enzyme. At very low substrate concentrations, when S is less than Km, the reaction rate is directly proportional to the substrate concentration. And this is said to be first order. So sometimes looking at a Michaelis-Menten curve, it is a little difficult to obtain the Km and the Vmax because of the gradual slope of the curve. So thanks to Hans Lineweaver and Dean Burke, we can rearrange the Michaelis-Menten equation to yield a linear equation and a corresponding graph called the Lineweaver-Burke plot. It's also called a double reciprocal plot because it plots 1 over the initial velocity, or V0, versus 1 over the substrate concentration. Let's see what this looks like. This line represents 1 over the initial velocity. This line represents the slope. The slope of the line is equal to Km over Vmax, where the line intercepts the y-axis, or 1 over V naught, is the intercept. And this is equal to 1 over Vmax. And where the line intersects the x-axis, And the x-axis, by the way, is 1 over the concentration of substrate. This intercept is equal to minus 1 over Km. And while you can see this is a little bit more complicated, it actually does make it easier to compare different enzymes and their affinity for substrate. So there are some other factors that can affect the enzyme activity. These include temperature, pH, and the concentration of the enzyme. Now let's move on to enzyme inhibitors. So there are 3 different types of enzyme inhibitors that I'm going to talk about. Reversible enzyme inhibitors, which include competitive and non-competitive inhibitors, and irreversible, also known as suicide inhibitors. Competitive inhibitors are structural analogs of the substrate. They bind to the active site of the enzyme, and importantly, they increase the Km for the substrate, but they have no effect on Vmax. We can see their effect on the Michaelis-Menten curve here, and you can see that it shifts the curve downward. Because these are competitive, they can be outcompeted with increasing amounts of substrate, so the Vmax doesn't decrease and the Km is increased. We can see that here. You can also see that on the corresponding Lineweaver-Burke plot. In the absence and in the presence of the inhibitor. And you see that where this line impacts and intercepts on the x-axis, or 1 over substrate concentration, changes in the presence or absence of the enzyme inhibitor. So once again, important consideration, competitive inhibitors bind to the active site, but they can be knocked off that active site by increasing the substrate concentration. Non-competitive inhibitors, on the other hand, bind to an allosteric site. They do not resemble the substrate, and as a consequence, they decrease the Vmax, but have no effect on Km. And you can see that here on the Michaelis-Menten graph. We can also see on the corresponding Lineweaver-Burke plot that where the line intercepts with the x-axis does not change. Finally, we have mechanism-based inhibitors, also known as suicide substrates. Suicide inhibitors bind to the active site and irreversibly inhibit the enzyme. The kinetic effects are very similar to what we're seeing with the non-competitive inhibitors. Before we move on, I want to give a couple pharmacological examples of enzyme inhibitors. The HMG-CoA reductase inhibitor lovastatin, which is used for hypercholesterolemia, is a competitive enzyme inhibitor, whereas aspirin, which is an anti-inflammatory, is a suicide inhibitor for the enzyme cyclooxygenase. Other examples include cancer drugs such as 5-fluorouracil and methotrexate, as well as drugs used for gout and hypertension. Now let's look at allosteric regulatory enzymes. Allosteric enzymes generally regulate rate-limiting steps and pathways. They catalyze irreversible steps, and they can be regulated by the products, which we see here. This enzyme catalyzes the conversion of B to C, but it's also regulated by its product, E. Allosteric enzymes can be regulated in many different manners. They can be regulated allosterically, that is, a molecule binds to a site distinct from the active site of the enzyme. They can be regulated covalently, such as by phosphorylation, or by changes in enzyme abundance. Key regulatory enzymes may be regulated by all of these mechanisms. So let's look a little deeper at this. The word allosteric literally means other site. As I mentioned, it's a distinct site from the active site of the enzyme. Allosteric effectors can either be activators or inhibitors, and by binding to a distinct site, they can change the conformation of the enzymes. In many cases, allosteric enzymes are multi-subunit. By binding to an allosteric site, these allosteric activators or inhibitors may ultimately influence the way that these multi-subunit enzymes assemble. A key difference between allosteric regulatory enzymes and the enzymes we looked at a moment ago is that allosteric regulatory enzymes generally display sigmoidal kinetics. This is probably because most of these are multi-subunit. Therefore, they do not obey Michaelis-Menten kinetics. If we were to look at a graph for an allosteric enzyme, it would look something like this. In the presence of an allosteric activator, we see that the curve shifts. There's a decrease in Km, an increase in Vmax, or possibly both. Whereas in the presence of an allosteric inhibitor, we see that the curve shifts down and to the right. Allosteric inhibitors generally increase the Km, decrease the Vmax, or both. Some common allosteric effectors include energy metabolites such as ATP, ADP, AMP, acetyl-CoA, or NADH, or as I mentioned a moment ago, end products of pathways, for example cholesterol or fatty acids or nucleotides. Finally, let's talk about isozymes. Isozymes are proteins that catalyze the same reaction but differ as follows. They have different kinetic properties or different regulatory properties, and isozymes often have different tissue or cellular localization patterns. Isozymes have diagnostic value, and changes in tissue-specific isozymes in the plasma can help identify damaged tissues. And we see a list of different isozymes in the table below and some of the major diagnostic uses that they're associated with. I want to focus in on two different examples from this table. Creatine kinase, which can be used to diagnose muscle disorders and myocardial infarction, and lactate dehydrogenase, which also has a role in the diagnosis of myocardial infarction. In the case of these two isozymes, they're multi-subunit. Creatine kinase is a dimer. It has two types of subunits, M or B, and three types of dimers, MM, MB, or BB, whereas lactate dehydrogenase is a tetramer, two types of subunits, H and M, and 5 different permutations of the tetramer, H4, H3M, H2M2, HM3, or M4. Importantly, the genes for these different subunits may be expressed differently in different tissues, and this can lead to variations in subunit composition. So let's take a look at these two graphs to see how creatine kinase could be used to help diagnose a myocardial infarction. We can see in the top graph, if we look at creatine kinase levels in the blood, we see two peaks. We see one peak for the MM dimer, and we see a smaller peak for the MB dimer. However, in a patient who's had a myocardial infarction, we see that the level of MB dimers is elevated, and this is because this isozyme of creatine kinase is expressed more readily in myocardial tissue. And following myocardial infarction, this isozyme is liberated into the blood.